Professor Jane Rudolph, Microbiology, final exam, ARQ number 2, helper T-cell stimulation. APC, macrophage, non-specifically presenting antigen with MHC2 with antigen on its surface, presenting to helper T-cell, specifically binding antigen, which has T-cell receptor and CD4. APC releases interleukin-1, which activates helper T-cell to release interleukin-2 and interleukin-4, which activates helper T-cell and stimulates two other pathways. Interleukin-2 plus co-stimulus from dendritic cell activates cytotoxic T-cell, which has T-cell receptor and CD8, so that it can take part in cell-mediated immunity. In the other pathway, interleukin-4 and interleukin-5 plus helper T-cell 2 binding activates B cells, which have MHC2 receptors and plasma membrane receptors like antibodies, so it could take part in humoral immunity. This is Professor Jane Rudolph's diagram. To help our memories, let's go over this a little bit slower this time. This ARQ number 2, helper T-cell stimulation begins with a macrophage and an antigen, and it's an APC or antigen-presenting cell which non-specifically presents the antigen, which you don't actually have to label for the ARQ, which means that it's not picky about what antigen it presents. The antigen is presented on the cell surface on the major histocompatibility complex, but we're just going to call it MHC2 here and note that it has an antigen, abbreviated AG, attached to it. A helper T-cell binds to the MHC2 antigen complex, and it is specific to the antigen, so we say T sub H specifically binding antigen. You have to draw the T-cell receptor, which binds to the antigen, and then also a little doohickey called a CD4, which is a glycoprotein that binds directly to the MHC2. The antigen-presenting cell then releases the cytokine interleukin-1, which activates the helper T-cell, and Professor Rudolph draws the little star and exclamation point to represent activation. It stimulates the helper T-cell to produce interleukin-2 and interleukin-4, which activates the cell further to proliferate and differentiate, as we learned in the first ARQ, but also goes along to stimulate two other pathways. In the first pathway, interleukin-2 plus a co-stimulus from dendritic cells activates the cytotoxic T-cell. As a T-cell, this cell also has a T-cell receptor, which this time we can label as TCR, and it also has a little doohickey similar to the CD4, but this time we call it a CD8 because it actually hooks up to a different histocompatibility complex, or MHC. This cell then goes on to take part in cell-mediated immunity. In the other pathway, interleukin-4 and interleukin-5 plus helper T-cell type 2 binding activates B cells, which will eventually directly attack antigen-bearing cells as discussed in ARQ number 1. The B cells have MHC2 receptors similar to the macrophage, but they also have plasma membrane receptors that are like antibodies and are actually sometimes called membrane-bound antibodies because structurally and functionally they're similar to antibodies. This B cell then goes to take part in humoral immunity. And that is it for this ARQ.